Much, but not all, of agroecology is associated with plants, whether we're talking about maize or tomatoes or sugarcane or what. Agroecology is about plants. Now, it's also about animals. Granted, we're talking about pigs and chickens, and sometimes it's about fish when we're talking about aquaculture and aquaculture combined with agriculture. But one of the foundational ideas has to do with the way plants grow. Plants really form a really important foundation for agroecology. So we're going to begin with the question of plant uh, plant physiology is what we usually refer to it, or ecological physiology of plants. There are three topics that we're going to cover that sort of summarize the whole thing. One is photosynthesis, second, respiration, and finally, allocation. The the process of photosynthesis is undertaken in these organelles that exist inside of the cells of plants. They're called the chloroplasts. Inside of the chloroplasts, you have a variety of different structures that we understand now to be involved in the photosynthetic process. Now, so photosynthesis basically involves two distinct activities. First of all, capturing the energy that comes in from the sunlight, and then second of all, allocating the carbon out of the air using the energy that comes in from the sunlight. Now, the first step in photosynthesis, the capturing of the energy, happens in the chloroplast, the thylakoid membranes inside of the chloroplast here, as indicated in this figure. And that's where a photon from the sun comes in, and that photon excites an electron, and the electron goes through a series of changes, which ultimately ultimately involves the production of energy compounds like ATP, for example. The ATP, NADH, those are the sort of the energy carrying pieces of the whole physiological process, and that's what we're going to use now to do the next phase of the process. That next phase involves the fixing of carbon, and that next phase happens not necessarily during the day. Of course, the first phase has to happen during the day because that's the only time sunlight operates. But the second phase operates sort of normally. Carbon is shunted into a biochemical cycle referred to as the Calvin cycle. And through the Calvin cycle, the carbon is stored mainly as glucose, the molecule glucose, which then therefore contains not just the carbon taken out of the air from carbon dioxide, but also the energy that's stored in the various chemical bonds inside of the glucose. When introducing the concept of photosynthesis, what's usually done is I usually present a very simple formula, the basic photosynthetic formula, where you have carbon dioxide plus energy plus water yields glucose and oxygen. Now, what's not frequently said right at the beginning is that process, that chemical process, is mediated by an enzyme called Rubisco, the most common enzyme in the world. Rubisco has a morphological form where it has binding sites on its surface, and those binding sites are specifically associated with the morphological structure of CO2. So CO2 fits in right to those binding sites like that. Now, the combination of CO2 and Rubisco is what we get that's where we get the glucose, and the glucose, the elements that are in that process recycle back there to in what's called the Calvin cycle. Occasionally, a molecule of oxygen, which appears sort of similar morphologically to the CO2, gets taken up by Rubisco. When that happens, the chemical phosphoglycolate is produced, and phosphoglycolate happens to be poisonous and not good for the cells. Now, why does that happen? Well, in the atmosphere, we have a lot of CO2, of course, but we also have a lot of oxygen. So oxygen is frequently there, and Rubisco occasionally just sort of does it. Now, a long time ago, CO2 was dominant in the atmosphere, and oxygen virtually didn't exist. Oxygen actually came to exist as a common element in the atmosphere only because of this process of photosynthesis, where you could say that Rubisco is the one that is responsible for putting all that oxygen in the air, which it then utilizes to produce this poison. This process is referred to as photorespiration, and it is just generally regarded as a basic inefficiency of the enzyme Rubisco, incredibly so because Rubisco happens to be so common, so old, and involved in every plant that engages in photosynthesis. So there's something kind of remarkable about the fact that the most common enzyme in the world and the one that's most responsible for this process of photosynthesis, which is so essential to absolutely everything that exists in the world, right now the way our biosphere operates, it operates on the foundation of this particular protein. It's remarkable that it's so inefficient at what it does, that oxygen can simply gum up the works as it does. Now, there's one way that plants have, some plants have dealt with this question in a, shall we say, a creative way. Evolution has been creative about dealing with this particular question when it's especially important to deal with. So plants have been relatively clever in dealing with, partially dealing with this problem. At least some of them have. Here you have, let me explain this. Here you have a model of a leaf cut out so you can see the cellular structure of the leaf. Up here you have the upper epidermis, here you have the lower epidermis, and you can see then below the upper epidermis you see the row of palisade cells, and then below that you see the mesophyll cells. And you'll notice the mesophyll cells aren't all that well organized. There's a lot of spaces between them, and that air gets into those spaces. That's a pretty important part. The tube over there is the trachea. That's where the that's where the various juices flow up and down into the plant. But the most important thing is here. This is called a stoma, plural stomata. But the stomata are very important for the plant. That's where the air actually gets in. Now, see where the mesophyll cells are. The mesophyll cell is where the photosynthesis is going on. Now, the photosynthesis begins with the capturing of light in the light reaction process. That's true. But then the Calvin cycle is automatically moving, constantly moving, and Rubisquist constantly doing his thing, grabbing a CO2, making a glucose, grabbing a CO2, making a glucose, etc. Occasionally grabbing an oxygen, which is the problem. So imagine what happens here now if you did not have that opening. Why, let whatever air you happen to have in the plant at that time, eventually, as the Calvin cycle was moving around and around, using up all the CO2, why the air inside of the leaf would have a high concentration of oxygen, which means that photorespiration would happen very, very frequently, which would be a huge problem. So the stomata are openings there, which means that the air is constantly coming in. But now the stomata, the way they're organized, a given stomata is like a hole like this, but each side of the hole has two what are called guard cells, like that. And these guard cells are like little balloons with a sort of thickening on the inside, so that they're like this when they're relaxed, and then when you fill them up, why they fill up like this, and so they open. And so the plant has control through the process of managing the turbidity of the guard cells. The plant can open or close the stomata. Now, the stomata are important for, as I said, for letting the air in, so you have oxygen, so that you have carbon dioxide that you can use in the whole photosynthetic process. But also, if it's open to the air, why, water can evaporate outside of it. That is, water vapor can escape through the stomata also. So the stomata present a solution to one problem, that is getting the air in for photosynthesis, but the generation of another problem, that is loss of water vapor. Now, this, of course, is going to be especially important in situations where water is a problem. So areas that are desert-like situations, or areas that are subject to droughts, periodic droughts, that's going to be a problem. And that's a problem that the plant will be water stressed if that happens. Evolution has produced for us a totally different form of photosynthesis. It's called C4 photosynthesis, and if you look at a plant that engages in C4 photosynthesis, you can immediately see a fairly dramatic difference in the structure, the microstructure of the leaf itself. Here, the trachea, you'll note, are surrounded by guard cells. The guard cells are distinct from the normal mesophyll cells, so the air still comes in through the stomata, but in a C4 photosynthesis, the Calvin cycle does not occur in these mesophyll cells, but rather occurs in the guard cells that surround the trachea. The guard cells, what I referred to as guard cells, are actually called bundle sheath cells. And the bundle sheath cells, as diagrammed here, is the place where the Calvin cycle actually happens. So basically what can happen is the process of photosynthesis can begin with the light reaction, and then chemicals are produced, chemical compounds are produced. Actually, these compounds have 4 carbons in them, which is where you get C4 to describe the process. And those are shunted into the bundle sheath cells, and the Calvin cycle operates inside of the bundle sheath cells. So what happens is the depletion of carbon dioxide in the air that's occurring around the mesophyll cells doesn't really happen inside of it's not relevant to what's going on inside of the bundle sheath cells. So effectively, you divide the whole process of photosynthesis into 2. It's the generation of the energy from the photons from the sun that happen in the mesophyll cells, but then that 4-carbon compound, which is basically storing the raw material for the Calvin cycle, goes into the bundle sheath cells. That's in the bundle sheath cells is where you get completion of the Calvin cycle. So this solution is actually a convenient, is an elegant solution. If you're concerned with the problem of water vapor loss, as you will be, for example, in very hot, dry climates, or when you're subjected to droughts, the C4 photosynthesis is an excellent way of avoiding the problem of photorespiration. So in sum, what you have is you have the CO2 and the oxygen, or the CO2 coming in through the stoma, and the oxygen and water going out from the stoma. In a C3 plant, what happens is that the Calvin cycle operates in the regular mesophyll cells as normal. The C4 operation on the right here, the carbon dioxide comes into the and oxygen also comes into the leaf, and in the mesophyll cell, these intermediate compounds are created that get shunted into the bundle sheath cells, which is where the Calvin cycle actually operates. Meanwhile, the oxygen leaves and the water leaves also, but having stored all of that C4 product in the bundle sheath cells, when things become too hot, when things become too dry, the stomata just can close for a while, so that the evapotranspiration, the evaporation of the water vapor from inside of the leaf doesn't dry out the leaf. It's an elegant solution. So it's at least interesting, perhaps important, to understand what's happening at the cellular level with photosynthesis and respiration and all that. But in agroecology, we're usually more interested in knowing what's happened at the leaf level, and even at what's happening at the whole plant level. Now, what happens at the leaf level or the whole plant level, similar basic qualitative relationships that go on, is this. And what we have here is we have on the y-axis we have net photosynthesis. This is that's important. And on the x-axis we have the amount of light that's coming in, the irradiance. Okay, you can see 3 important points here. Well, first of all, you see where the 0 point is. The actual curve goes down below 0, and that's really quite important, okay? Because your net photosynthesis sometimes is negative because you have a certain amount of maintenance that the plant has to provide for itself, replacing root tissue, replacing leaf tissues, growing new leaves, etc. So there's a maintenance cost, and that maintenance cost is given, as you can see right here. Then there's what's called the compensation point. The compensation point is a point at which there's just exactly the amount of photosynthesis going on as what's required to maintain it. So your net photosynthesis is actually equal to 0. So that's the level of light at which you're perfectly balanced between what it costs to keep the plant going and the energy that the plant's getting from photosynthesis. And then finally, you have the light saturation point, which is the amount of light at which the plant has maximized its photosynthesis. So increasing light beyond that doesn't increase photosynthetic intake at all. And here we see one of the most important elements of the difference between C3 and C4 photosynthesis. On the graph on the left, you can see which is 35 degrees C. C3 plant has a compensation point which is much larger than the C4 plant, and it saturates at a much lower level of irradiance, a much lower light level. C4, on the other hand, increases dramatically, and this is typical of C4 plants. They respond to increased light frequently without any kind of light compensation point at all. Now contrast that at a lower temperature, at 10 degrees C, for example, and you can see the situation really differs. What you have, there's two possible curves for C4, obviously, but see the way the C3 curve, the way the C3 photosynthetic curve intersects C4, and you can see at a lower temperature then, at a lower temperature with higher irradiance, while you can have a situation where the C3 plant actually outperforms the C4 plant. So the temperature actually makes quite a bit of a difference. Associated with the temperature is frequently the humidity, and I'm not going to show you separate graphs for the humidity, but you can imagine what happens with these curves when you're talking about different levels of humidity. In this world map, we see the distribution of C3 and C4 grasses around the world. And, you know, it's exactly what you would expect. The C4 grasses tend to be in the tropics, and the C3 grasses tend to be in the temperate zone. It's precisely exactly the kind of thing that you would expect. In the tropics, you have very frequently you have drought. You have very dry situations in the dry season. A lot of rain in the rainy seasons, that's true, but the drought in the dry season, and the temperatures are really quite high. At high temperatures and less water conditions, that's when C3, C4 plants are favored. And exactly, if you look at the distribution of grasses, C4 versus C3 grasses in the world, that's precisely what you see in this map. Now, stop to think of the way a plant operates, what a plant's life is about. Okay, you have the leafy material on top, the green material on top, and then you have the root material on the bottom. And what happens there is you get energy on the top, and you get your nutrients on the bottom. Now that's very different from what we do, right? I mean, we, Homo sapiens, we have one organ that takes in energy and material. So a plant has material from the leaf material, the shoot, the above ground stuff, but it gets its energy from the above ground part, but it gets its nutrients from the below ground part. There's a distinction between then the energy and basic material capture from the above and the nutrients that are necessary to engage in that process from the bottom. So it's sort of like gaining energy and gaining the materials to utilize that energy in two different organ systems of the plant. So that sets up a question that animals simply don't deal with at all, but plants really have to deal with, and that is the allocation of the materials that you get from photosynthesis. Where do you put it? Do you put it into more leaf growth or more shoot growth? And that's a classic question that plant physiologists have been concerned with for many, many years. So if you're a plant, what do you do? What do you do with the photosynthate that you get, the stuff from photosynthesis? Do you allocate material to grow more leaves and more shoots so that you can do more photosynthesis? Or do you allocate more to the root system so that you can find more nutrients that you need for the basic life processes? It's a fundamental problem. As I said just a moment ago, it's a fundamental problem that plants face that physiologists have been dealing with for quite some time now. Consider this plant growing on a hillside right here, these plants growing on a hillside right here. Well, you see the water comes in and the water is going down, and the water eventually filters way down to the bottom. The plants are growing, and they're annual plants, so basically they're not going to put a lot into the root structure because they're not concerned about next year, they're just concerned about this year. So they just want those roots to get down there enough so they can get enough water so that they can grow, and enough nutrients so that they can grow, right? Now contrast that with a perennial plant. A perennial plant has to be concerned with not only putting on the photosynthetic material and the root material for this year, but has to worry about next year also. So all of that water that comes down to the lower levels then, the perennial plant is able to put more roots down to get at that water. So you can see the difference here between the allocation strategies. In this plant, the annual plant, the allocation strategy would be to concentrate on more photosynthesis, more photosynthesis. Of course, you have to put something in the roots, but the allocation strategy in the perennial plant is we have to allocate for this year, but we also have to be concerned with next year. Sometimes evolution sort of goes wild, and that allocation becomes very, very important for a perennial crop, sometimes for an annual crop too. But it goes wild with the allocation of materials to the root material itself. Sometimes these tubers they are there for the storage of energy for next year, but also sometimes they're used in the dispersion of the plant itself, in the natural ecosystem, I mean. But for agroecosystems, obviously one of the allocation issues that enters into the equation here is the production of tubers, which are agricultural products. Another issue that has emerged recently as a really kind of hot topic in agroecology is this question of perennialness versus annualness. Most of our grain crops are annual crops, of course, but there's a lot of people, especially at the Land Institute in Kansas, that are talking about the perennialization of some of the grain crops. And this slide is really quite amazing. This is showing the natural grass, wheatgrass, and comparing it to the actual the growth of domesticated wheat. And look at the root systems there. Now those root systems are, on the one hand, they're allowing the grass itself to be perennial because they're putting a lot of storage down under the ground. But look at the amount of the ecosystem that they're, or the amount of the environment that they're actually sampling. All of that water and all those nutrients that go all the way down to the deep layers of the soil, that's really taking advantage of space in the ecosystem where they can grow. Restricting that area to domesticated wheat, as would be implied by these pictures right here, you're restricting yourself to very, very limited potential of what exists in that system. This experiment is really kind of, in my mind at least, is a classic experiment illustrating the way plants actually do decide how to allocate their various how to allocate the photosynthate, that is the material that comes out of photosynthesis. Here you have two varieties of the same species. You have Swiss chard and sugar beet. That's exactly the same species. They're just genetic differences there that create two different agricultural products. Obviously, one, the sugar beet, obviously allocates an enormous amount of energy to the root system. The other, Swiss chard, does not apparently allocate that much energy to the root system because it's concerned, I say we bred it so that it's concerned with, to the leafy material, which is what we eat. This is the basic structure of the experiment. What you did was you took the leaves or the shoots from Swiss chard, and you put them onto the rootstocks of the sugar beet, and you took the leaves from the sugar beet and you put them on the rootstocks of the Swiss chard. What then were the results? Well, the results were really quite spectacular. Here you see on the slide what the results were. Exactly what you would expect to happen if, in fact, the plant itself is making decisions about where it should be allocating its energy. So we see here how there's a sort of an internal genetic structure that the plants have. I keep saying decide. Well, the plants don't have a brain to decide anything. Obviously, these are genetic constructs which are dictating the way allocation should happen. And you can pretty clearly see from this experiment that you can even manipulate how that allocation strategy is actually plays out. Now, it's pretty clear that allocation of materials, and there's costs associated with it, especially if you're allocating things for storage. Allocating things for storage means that you're not going to be immediately producing leaves or roots to engage in the process of growth and eventually reproduction. There are direct costs, and these costs are associated with translocation. They're associated with chemical conversions, particular chemicals that you have to make. The concern with construction of special tissues, if you have these tubers that are maybe need a special kind of epidermis or whatever. But then you also have opportunity costs. Those opportunity costs are obviously you have to waylay the production of more photosynthetic material to produce even more stuff. So there are agricultural, agroecological reasons to have to be concerned with the storage activity. There are products, of course, that we normally consider in agriculture, such as potatoes, such as cassava. And here's a picture of a really interesting crop. This is called jicama, and as you can see, it produces both fruits and a root. The root is actually very delicious. So that's a very brief summary of some of the issues that I think are important associated with physiological ecology of plants that we need to understand for agroecology. Here's a quick summary of some of what I think are the take-home messages.